All right, welcome back. So we're going to talk a little bit more about the nephron. This will be the last one with the nephron, and then we'll talk about just the last bit of the urinary system, which is really just kind of the tubing, the storage tank, and the rest of the tubing. So again, the urinary system, the whole idea here is to get rid of cellular waste. Again, kind of like the garbage men picking up the garbage from the road so we don't get a bunch of garbage in between our house and the road and it doesn't back up. It also is going to get rid of certain hormones and other metabolites and things that you might have ingested that end up in the blood that you don't want to keep in there. Again, we've talked about how a lot of this is started with the filtrate, and then we are going to reabsorb and secrete things into that tubule along the way. Again, the biggest thing here is getting rid of those nitrogenous wastes. The urea is probably the main component here. We're going to see that we secrete a lot of this out, but we do actually reabsorb it and use it to help us reabsorb some water. Same thing with uric acid. Creatine we're just going to be getting rid of. Urea, of what we generate, about 50% is excreted in the urine, and some of it is actually part of the interstitial fluid of the kidney to help cause reabsorption of water. So we have what's called some urea recycling on that. Again, a lot of our stuff here, in terms of those drugs and other stuff like that that end up in the urine, it's going to be secretion in that proximal convoluted tubule. Things like penicillin and other stuff like that. Other hormones also getting secreted out there. So like with pregnancy tests, HCG is actually excreted in the urine, and you can actually see that with an antibody test. So with the kidneys, like I was saying before, we do have this interstitial fluid gradient where the outer part of the kidney is less salty, and as you get more to the interior, it becomes progressively more and more salty. We're going to see we can use this in both the nephron loop as well as the collecting ducts to move and pull more water out. The collecting ducts, it's going to be when ADH is present, but we can also do this a little bit in the nephron loop, especially in those juxtaglomerular nephrons that go way down in. So what we have going on here in order to kind of maintain this as well as reabsorb more water out in this nephron loop is what is called a countercurrent multiplier. Again, this helps establish that salt gradient, and we have these differing permeabilities with the water and salt depending on the ascending or descending limb. What we end up seeing in this one, we can get the water to move from the tubular fluid to the interstitial fluid and then get taken back into the blood vessel, and we can use this also to retain a lot of the salt. So the image I'm going to show you here is kind of going to show this. What we have going on here though is, like I said, that ascending loop, it is not permeable to water, but what we have going on here is we have a lot of pumps that are actively pumping salt out. What this really means is there's more salt at the bottom of that nephron loop in these juxtaglomerular nephrons. And what this means is you're going to get a salt concentration that is most salty the furthest down in the medulla. And as you work your way up, that gradation is going to change a little bit. So and this maintains that salt concentration. And you can kind of see this right here. So we can see here is this nephron loop right here, or the loop of Henle. And the descending loop, we're going to see it is impermeable to salt, but water is able to leave. But let's start with the ascending loop. The ascending loop here, you can see down at the bottom, this fluid is rather salty, so 1100 milliosmolar. You can see as it starts working its way up, it gets a little bit higher, 1200 milliosmolar. Then you can see we start pumping the salt out. Again, more salt here. You can see we get the osmolarity of the medulla at these levels right here. So as we pump up, there's less salt to pump out, so we get a less of a concentration. But you can see this gradation from the medulla from about 300 milliosmolar to about 1200 milliosmolar as you work your way from the outer medulla to the inner medulla. The reason why we call this a countercurrent multiplier, or countercurrent mechanism, is again, we pump all this salt out on the ascending limb, but it's impermeable to water. So we pump the salt out, it can establish this gradient. But on the descending limb, in order to actually have this work out, you have, let's say it's about 300 milliosmolar fluid in terms of salt right here. As we start approaching this outer medulla, you're going to see, okay, we're going to have this permeable to water here. So through osmosis, if it's saltier out here, water is going to leave the tubular fluid to go out to the interstitial space. We have this gradation. So when it gets to right here, let's say this is 300, this is 301. In terms of the osmolarity, water is going to leave. Well, that's going to make this more salty in here. But as that water moves down here, this is now going to be more salty on the outside. So what happens is, because of the salt gradient, as this tubular fluid gets saltier, we still have a higher concentration of salt on the outside in the interstitial fluid. So we continue to pull water out all the way down this descending limb. We get to the bottom, then the permeability changes. We pump the salt back out to maintain that gradient. We'll get into what the Vasa recta is doing to pick up the water and salt in a second here. But the main thing is, this ascending limb maintains the gradient and sets up this countercurrent mechanism for the water to be pulled out all the way down. And then you can see as we get to the back up to the top here, we've pumped out actually more salt than it was to start with. So the fluid in here is now way less water in it. There's actually less salt in it, but we've concentrated the waste products. If ADH is present and we get into this collecting duct, now we have to go through that medulla again, and we have a fluid on the inside here that is not particularly salty. So if ADH is present and there's aquaporins present, as this water can now as this tubular fluid travels back down the collecting duct through the medulla, we now have again a countercurrent mechanism where we can pull water if we need to. The other thing we have going on here is about 50% of that urea is going to be pumped. About 50% of that urea is going to be pumped out, and that also creates a gradient to draw the water out into this area. That gets pumped out of the collecting duct here, gets taken back into the ascending limb. So we're going to get this little urea recycling that's going on between the collecting duct and the loop of Henle. But again, having more dissolved substances in that interstitial space means we're going to draw more water out. What's kind of interesting on these juxtamedullary nephrons is they have that Vasa recta. That was that blood capillary that was a little bit different than the paratubular capillaries. On these juxtamedullary nephrons, we're going to see the blood flow on these ones is actually traveling in the opposite direction of the tubular fluid. So if you have the descending limb and the ascending limb, you're actually going to have the Vasa recta going down when the fluid's coming up the ascending limb. It's going to loop down around the bottom and come up the descending limb on the other side. What this helps us to do, and we'll see it on the next one here, is it helps to get both the water and salt back into those blood vessels. As you travel down next to that descending limb, there's a lot of salt present in the interstitial fluid. A lot of that is going to diffuse into the fluids in those blood vessels. What that actually does is then is going to set up a gradient for when it gets up to the other side there where the water is getting pumped out. It's going to set up a gradient to draw a lot of that fluid then back into the blood vessel. And the water goes in and the salt goes back out. So you can kind of see it right here. On the way down, so here's the loop of Henle. The flow is traveling this direction. You can see the Vasa recta is traveling the opposite direction. As it comes down this descending limb here, it's going to lose some water out here and it's going to pick up some salts. But as it gets to the bottom here, we're going to see now, because it's picked up so much salt, a lot of water is going to be traveling back in here. We're going to lose a little bit of salts back out here. But what this really means is we've been able to reabsorb most of our water and salt and therefore maintain our blood volume by having this loop around the opposite direction of that tubular fluid. So this is all going back in the bloodstream, which is going to maintain our blood volume, maintain our blood osmolarity, and we're happy on that one. Like I said, the urea recycling contributes to this. About half of the urea that ends up in the urine is going to get pumped out in the collecting ducts. It then diffuses back into the ascending limb based on concentration gradients. So it's cycled through there. But again, this creates a osmotic pull for water to leave the tubular fluid and can help with that reabsorption. Like we said, after filtration, it's either reabsorption or secretion, and that's really what's going to determine what ends up there. By the time you're getting to the bottom of that collecting duct, what you have at that point is going to end up being urine. And again, mainly water, some dissolved substances in it, a few waste products. Again, goes into that renal papilla, into those calyces, and is going to get out to the rest of the urinary tract at that point. So proximal convoluted tubule is doing most of the reabsorption, almost all the nutrients, most of the water, most of the ions, a little bit of secretion of some drugs and waste products. As we get to the Vasa recta and the nephron loop here, more reabsorption of water and some of those sodium, salt ion components. It helps you get that gradient where you have a much saltier interior of the kidney versus the less salty exterior cortex. When we get to the distal convoluted tubule and the collecting ducts, definitely sodium reabsorption if aldosterone, or depending on aldosterone or adrenonatric peptide, water reabsorption depending on aldosterone or ADH secretion. Again, how much potassium we're reabsorbing is going to be somewhat dependent on what the signaling is there. Calcium reabsorption if we have parathyroid hormone present. And again, depending on pH, we may be excreting or retaining acid or base with these cells as well. If you've ever heard of something called a diuretic, what are these they're doing? One of the things they can do is actually help control blood pressure, because diuretics, they let you form more urine. They basically cause you to form more urine. So things like caffeine can act as a diuretic. There's other things that can also act as a diuretic. Main thing that these are going to do is reduce blood volume, which will reduce blood pressure. So these are, again, one of those many blood pressure meds that we've talked about. So that's part of the reason when you hear antidiuretic hormone, you know it's the opposite of this. So again, urine results from that filtration, absorption, and secretion activities. Most compounds are being excreted into here that are waste products. We're kind of trying to concentrate those and make sure stuff that we don't want, so certain metabolites, certain nutrients we don't want to become part of the urine are fully reabsorbed. Really, when it comes to the urine on this one, it really, what ends up in there depends a lot on gradients and other things like that. Urine, compared to a lot of other body fluids, is a sterile solution, at least it should be if you don't have any type of infection. So slightly yellow in color, partly from these urobiligens, which are a part of breaking down some of the byproducts of hemoglobin. So like I said, your urine is a product of filtered and processed blood plasma. Unless there's an infection, a kidney infection or a urinary tract infection, it should be sterile. Mainly water, some electrolytes in it, some hormones and drugs, small amounts of ketone bodies and things like that. But for the most part, mostly water. Again, depending on hydration status and other stuff like that. One of the things that we normally talk about in lab is specific gravity. Specific gravity of 1 would be the same as water. Urine, we're going to see, usually has a few more dissolved substances, so it's usually 1.003 to 1.030. So it's usually a little bit more stuff going on in there than just straight water. Things that shouldn't be in urine though, glucose, ketones, more than trace amounts of protein. We shouldn't see blood cells, we shouldn't see hemoglobin. All that could indicate something is wrong with the kidneys or wrong with kidney function or blood pressure. We normally, like it says there, about 1 to 2 liters a day. Again, you're going to be very dependent upon fluid intake and other ways you're losing water. This is the one way we can really regulate water loss. So if you're doing something that is causing you to lose water, sweating, not consuming much water, if you're not bringing much in, again, this is how we excrete outgoing water. So if we're not bringing more in, we tend to try to conserve more. Like it says there, it's about ½ liter a day to make sure you're getting enough to get rid of waste products in the body though. So if you're not doing that, you are going to have a buildup of waste happening if you're not getting rid of at least about ½ liter of urine a day. And again, all these factors that can influence this, and again, a really easy thing to ask in like a little matching or a thing like that. What can decrease urine volume? What would increase it? And again, a lot of these things are also messing with that GFR rate, that glomerular filtration rate. Generally, the higher the rate, the more urine formation. The lower the rate, the less urine formation. And you can see some of the things that we talked about affecting GFR. They also affect urine volume in terms of ADH increases or decreases in that, what it does to urine volume. Same thing with adrenonatric peptide. But simple sense, we try to maintain water balance. So if you take more in, you tend to get rid of more. If you take less in, you tend to get rid of less. If you're losing water in a different way, you tend to try to conserve more in the urine. So again, homeostasis. pH should be slightly acidic to, can be sometimes even slightly alkaline, but tends to be on the slightly more acidic side, usually a little under neutral. Again, depends on what your diet is more than anything. A lot of proteins, wheat products, things like that tend to give you more acidic urine. More fruits and vegetables, with the exception of citrus fruits, tend to give you more alkaline urine. So really dependent upon what's going on there. Again, specific gravity is the density of the urine compared to water. It should be slightly higher due to the salt and creatine and a few other things that are in there. Again, greater hydration will get you a lower specific gravity. If you're dehydrated, your urine tends to be more yellow or darker in color. It will also tend to have a higher specific gravity. Again, normal range 1.003 to 1.030. Like I said, it should be slightly yellow. I mean, if you're getting really clear urine, you're kind of probably overhydrating slightly, but you don't want a super dark urine either. Again, can change depending on what you've eaten and other stuff like that. It should be clear. If it's cloudy, it could mean protein or bacteria or other stuff in the urine. And this is always the slide that makes me laugh because the smell is urinoid. It smells like urine. Shouldn't have a fruity smell. Shouldn't really smell ammonia-like. That would mean bacteria are usually breaking it down. A lot of times, if you've ever been around a cat litter or a house where cats have urinated a lot of times bacteria will break down the urea in urine and give you more of that ammonia smell. If it sits a while, that's what happens. It breaks down a little bit more and you get ammonia. Fruity smells or maple syrup kind of stuff there, those are certain diseases that kind of give you those odors and smells. So really the last thing we have to do after this now is we'll take a look at the rest of the urinary tract and then we'll be on to the reproductive system and then that will be it.